Roots are the source of life for all plants because they connect the plant to the water and nutrients that the plant needs in order to develop into a healthy adult. The process of root growth depends on a complicated balance of three types of cell development zones. A typical root tip consists of a protective root cap. Meristem zones focus on division of the rapid growth of cells. Expansion zones focus on stretching and moving cells in a particular direction. And differential zones focus on assigning new cells to their roles in the plant. All three zones have to work together in a balance in order to create a stable plant structure. But how do these zones know when to activate and when to stop? How do they keep the balance? The answer is in plant hormones. Much like the hormones in humans, plant hormones trigger developmental growth within the plant and help tell the cells when to activate and when to turn off. The most well-studied plant hormone controlling root growth is auxin. To understand how auxin works, let's look at a transcription factor called plethora transcription factor. Plethora transcription factor is a kind of protein that is only transcribed in stem cells at the tip of the root and becomes less and less as the cells divide. According to a research conducted in the Netherlands, the plethora transcription factors only bind to the DNA in response to auxin accumulation. The more auxin in the cell, the more often plethora will bind to the DNA. When there is an abundance of plethora present, the more genes related to cell division will be transcribed and the cells will divide rapidly. When there is less plethora present, more genes related to cell growth and differentiation will be transcribed and the cells will not divide. Elongation and differentiation processes will instead take place. Plethora is under control of and serves to the hormone auxin. Plethora induction requires prolonged high auxin levels, and the auxin gradient is formed in response to a plethora concentration gradient. Auxin is able to read signals from plethora and regulate the rate at which the cells divide, elongate, or differentiate. The two work hand in hand in order to balance plant growth. Auxin acts as a signaling chemical to tell the meristem when to activate cell division. Think of auxin as a traffic light that tells which cells to grow and when. In response, plethora is like a traffic cop that can see the light and be the mediator between the cells. Auxin and the plethora transcription factor are important in the formation of secondary roots and adventitious roots. Adventitious roots are the reason that a branch or leaf can be cut from one plant and generate into a new plant, a phenomenon known as dedifferentiation. The origin of this important hormone has struck an argument between scientists for years. It has been theorized that the hormone auxin originates from the initial plant shoot, then moves to the area that controls lateral root development. This theory was recently concluded in an experiment where auxin transport inhibitor, naphthalamic acid, was injected into several positions of the root with results of less abundance and density of lateral roots. This gives strong evidence to the theory that auxin hormones are synthesized in young leaves and cotyledons, then move to control lateral growth. Once the signals are originated, they move up through the plant via cell-to-cell interactions and signaling. Hormones are signaled into the cell through influx transporters and out of the cell through efflux transporters. Scientists were able to genetically modify soybean plants by using a plasmid method to insert meristematic tissues with CRY1AC, a gene that deters certain insects. The result was a new generation of soybean plants that are resistant to these insects and have an overall higher survival rate. In addition, this new strain of soybeans is resistant to imidazoline-based herbicides due to the editing of the soybean's meristematic tissues with an inserted plasmid vector, PAC321. Scientists were able to insert a mutation called CSR12 into the plasmid vector, and this caused the soybean's meristem to produce an enzyme known as acetohydroxyacid synthase large subunit ATAH-ASL. This enzyme is what is responsible for the plant's ability to resist the effects of herbicidal treatments. These scientific advancements lay a foundation for commercial crop success in the future, as well as ecological stability. Because versions of each plant that are better fit for survival can be created using the most basic cell forms, crop industries and ecological communities will greatly benefit. Not only will this generate profit, but it will also help potentially keep plants from being at risk of disease, predators, and ultimately, extinction.
created using Powtoon.